In open darkness, a luminous line extends beyond the camera frame and continues through black water. It resembles one impossible animal, yet its length is built from thousands of specialized living units. Siphonophores are colonial cnidarians, relatives of jellyfish and corals, organized as integrated drifting bodies. Some can exceed the length of large whales, although extreme estimates depend on incomplete underwater views. Their mystery is not merely size, but how repeated modules cooperate without becoming separate individuals. The colony begins from one fertilized egg, preserving a single genetic origin across its expanding architecture. Through budding, it produces zooids that remain physically connected and assume different biological tasks. If you approached the ribbon, apparent repetition would resolve into feeding, defensive, reproductive, and swimming structures. No zooid represents the entire siphonophore, and most cannot survive independently from the colony. Length emerges through multiplication rather than enlargement of one conventional body. This developmental strategy lets delicate tissue occupy a vast hunting volume with comparatively little dense material, yet it also exposes the colony to tearing, turbulence, starvation, and misleading measurements. The investigation follows growth from a tiny founder to a coordinated chain longer than familiar giants. Every new zooid adds function, spacing, and potential reach while increasing dependence among parts. The whale comparison is visually powerful, but colonial organization is the real explanation. Siphonophores belong to Hydrozoa within the phylum Cnidaria, whose members possess stinging cells called cnidocytes. Many species inhabit the open ocean, including deep layers where sunlight weakens or disappears. Their forms vary widely, so conclusions about one genus should not automatically describe all siphonophores. In many groups, a stem carries repeated sets of zooids arranged in characteristic patterns. Some colonies possess nectophores, swimming bells that pulse and propel the connected body. Others rely more heavily on drifting, buoyancy, or subtle control of position. Feeding structures called gastrozoids capture and digest prey delivered by specialized tentacles. Reproductive zooids produce gametes, separating that task from routine feeding or propulsion. This division of labor resembles organs within one body more than a gathering of cooperating strangers. Nutrients move through connected gastrovascular canals, linking capture at one location with demand elsewhere. Nervous coordination also passes along the colony, though no centralized brain directs every response. When mechanical or chemical cues reach tentacles, cnidocytes discharge microscopic structures into suitable prey. Tentacles contract, drawing captured crustaceans, or fish toward gastrozooids for digestion. That cue-action adaptation sequence converts broad spatial coverage into concentrated nutrition for the colony. Prey encounters need not occur near the swimming bells or reproductive structures they ultimately support. The stem, therefore, functions as both scaffold and distribution route. Repeated modules can be added as growth zones bud new zooids in an ordered sequence. Older and younger parts occupy different positions, recording development along the living axis. Unlike a whale, the colony lacks a single muscular trunk enclosing all major organs. Its narrow, water-rich tissues require less mass per unit length than a vertebrate body. Becoming longer is therefore mechanically and energetically different from making one animal proportionally enormous. The surrounding seawater supports the colony, reducing the structural demands faced by giants on land. Neutral or near-neutral buoyancy allows length without heavy bones, though currents can impose dangerous strain. Direct measurements remain challenging because fragile colonies rarely reach the surface intact. Deep vehicles provide video, scaling lasers, and environmental context without hauling the organism through pressure changes. Even then, the camera may capture only a curve, coil, or partial segment. Estimating total length requires tracing the full body and knowing its three-dimensional position. A colony extending outside the frame cannot be measured confidently from a single image. Apparent scale also changes with lens geometry and distance from the vehicle. Consequently, spectacular sightings are observations of remarkable size, not automatic records. Species identification can be equally difficult when diagnostic zooids remain invisible or damaged. Some giant deep colonies belong to the genus Apolemia, known for long, complex forms. However, attaching that name to every immense spiral without adequate features would exceed the evidence. The longest credible values should therefore be treated as estimates with explicit uncertainty. If you watched a survey video, the unending passage might feel precise while the unseen endpoints remained unknown. One hypothesis predicts that longer colonies intercept more prey because tentacles sample a larger volume. The benefit should appear as increased encounters, provided prey density repays added tissue maintenance. Another hypothesis emphasizes patchy environments, where extended arrays bridge small differences in prey distribution. A cost hypothesis predicts greater breakage, where turbulence or shear increases. Siphonophores can contract sections or alter posture when disturbed, reducing exposed span. Swimming bells may pulse together, translating distributed signals into coordinated movement. The resulting turn or withdrawal can move vulnerable feeding structures away from contact. Coordination is not perfect rigidity. Flexibility lets the colony bend rather than resist every current. That flexibility complicates measurement while serving survival in moving water. Fragments may sometimes persist, but survival and regeneration differ among taxa and colony regions. It is unsafe to assume that any broken segment readily becomes a complete new colony. Growth observations must distinguish normal budding from repair after damage. Laboratory studies reveal development, yet many deep species cannot be maintained under natural pressure and feeding conditions. Field images reveal posture, but rarely follow one colony long enough to calculate lifetime growth. Length also changes dynamically as the stem contracts, extends, coils, or trails behind swimming bells. The same colony might appear radically shorter during contraction without losing zooids. Thus, a measurement is a moment in behavior, not a permanent anatomical constant. Long form carries ecological consequences by spreading predation across multiple small prey patches. Tentacles create a suspended capture field that can influence zooplankton and small nekton. When prey touches a tentulum, Discharge, adhesion, contraction, and transfer follow in rapid sequence. Specialized structures reduce the need for the whole colony to chase one target. This permits ambush feeding, where darkness and sparse prey favor extensive, low-mass traps. Bioluminescence in some siphonophores may deter attackers, lure prey, or serve other functions, depending on species. Observed light proves emission, while its exact receiver and purpose can remain uncertain. Some species produce striking red luminescence, unusual because red light travels poorly through seawater. Functional interpretations require behavioral tests. Rather than assuming every glow attracts food, you can regard the colony as a moving ecosystem interface, but its zooids share one developmental identity. Small organisms may shelter around it, predators may bite it, and prey become nutrients flowing internally. Carbon captured from mobile animals eventually enters waste, respiration, reproduction, or other consumers. When tissue sinks, it also transports organic material toward deeper communities. These pathways connect extraordinary length with ordinary ocean processes of predation and recycling. Whales concentrate enormous mass into powerful bodies that migrate, dive, breathe, and generate heat. Siphonophores distribute fragile modules through supported water, solving a completely different engineering problem. Calling one larger than a whale is meaningful only when the compared dimension is stated as length. Mass, strength, lifespan, and ecological impact do not follow from that single measurement. Future evidence requires complete, scaled recordings of undisturbed colonies from end to end. Multiple camera angles could reconstruct three-dimensional curves instead of flattening them into uncertain lines. Repeated passes must avoid slicing the body visually or physically with vehicle movement. Genetic sampling, when carefully obtained, could connect extreme forms with securely identified species. Long observations could measure budding rates, contraction ranges, feeding success, and damage under different currents. Flow sensors could test whether long colonies occupy calmer layers or actively change posture in shear. Evidence for prey interception would strengthen if encounter rates rose predictably with deployed tentacle area. Evidence for patch bridging would require mapped prey distributions around the complete colony. Breakage records could reveal whether exceptional length is restricted to unusually gentle conditions. Developmental data could show how long growth continues and which resources set its ceiling. No camera has yet turned every spectacular deep sighting into a fully measured biological biography. The unknown maximum should remain unknown until endpoints, scale, identity, and posture are established together. If you return to the luminous line, its greatest wonder is coordination repeated across living distance. Each zooid adds a task, each connection shares resources, and the colony lengthens without becoming a whale. A siphonophore exceeds familiar dimensions through colonial growth, while its true maximum remains a measurable scientific question.